region in Ontario, Canada, and I'm going to talk to you a little bit today about a joint project we have in China as well as at Wilfrid Laurier and Waterloo University that's looking at multitrophic interactions in a vineyard that's exposed to different amendment strategies. So just to kind of put things into perspective, I think we're all very aware of how our past and present management strategies in agriculture have led us down a path of non-sustainable soil degradation, plant degradation, communities that are not as resilient to environmental change. And we do need, with our growing population in the globe, to look at different management strategies to enable us to supply for future generations. So how can our understanding of ecosystem ecology help us to implement sustainable management strategies in agriculture? Because an agricultural field or orchard or tree plantation is still an ecosystem, albeit a very heavily managed one. So when I'm talking about ecosystems, I'm talking about paying attention to the multitrophic interactions that happen on both an abiotic and biotic scale. So for an agricultural system, we're looking at things that are already there. Things like soil, weeds, beneficial pests, inverts, microbes. And we're also looking at inputs that farmers or whoever's managing the land is trying to do to grow crops, to maximize their production with soil amendments, often chemicals, as well as cover crops, which is not a new concept, but something that we could really begin to focus on to make a more diverse and strong agroecosystem. And all of these elements, all of these components interact with one another in various ways. And if we do one-to-one component, it often has a cascading effect on other connected components in the system. So I'd like to introduce you to our Ontario-China Research Innovation Project that we fondly call OCRIF. And what it is a project that's funded by the Government of Ontario, and we're looking to combine two different research areas, Fujian Province in the left-hand picture here in China, and their tea plantations, which is a perennial system, and we're looking in the Niagara region in Ontario, here in North America, at vineyards, which is also a perennial system. So similar in their needs in terms of long-term impacts and responses, but a little different in different parts of the world, obviously. So the objective in our Niagara area was to determine vineyard system responses to the following ecological amendment strategies. The first being an agromineral known as Spanish River carbonatite, which is taken out of an area in north central Ontario, that also has been shown to have a symbiotic relationship with a lot of microbes, which can translate into things like nitrogen fixation in the plants that are present. And we wanted to measure this against traditional chemical inputs that we find in our vineyards in the region. The second strategy we were looking at was cover crops, which again is not a new concept, but what we wanted to do, instead of looking at the monocultures of the traditional, say, annual ryegrass, which we find a lot of in the Niagara region, we wanted to change it up a bit and create some more diversity. So we were looking to create a cover crop that was actually an intercrop of chicory, oilseed radish, alfalfa, and red clover. So we could really maximize the different areas of potential of all these crops. So a lot of resource partitioning to try to improve things like soil structure as well as the nitrogen balance. So for project implementation, we looked at two different sections. The first thing was a controlled condition in the greenhouse, which looked at the interactions and growth potential of the cover crop selection. So we're putting them together. How are they going to respond to one another in terms of competition, for example, as well as their growth potential overall when exposed to an agromineral as compared to the chemical fertilizer. After this, we brought out into the field and into common practice our strategy to see how other elements, such as invertebrates, weather, farming management strategies, were going to affect our treatment applications. So just to share a few of the results that we have so far in the greenhouse, this is just a visual of 30 days of growth in the intercrop setting between the fertilizer and the SRC. And while we don't see a huge difference, except for the oilseed radish there on the right, which you can see is a little more robust, it was actually kind of interesting to us still because there was no real big difference in growth between the two, and that's kind of what we were going for. It would be great to see more growth, but we're pretty happy seeing no changes between the two treatments. So to look at growth over time, we measured things like plant height, leaf count, percent cover, and in the case of alfalfa, red clover, all of them actually, this is just alfalfa here, the heights as well as the other growth parameters were very similar between fertilizer and SRC. Again, which is maybe not exciting, but to us it was kind of exciting because we have similar responses to the two. So by the end we were quantifying at harvest time the dry weights, and we looked at shoots and roots individually, differently, and we had similar responses. We did see a significant difference in our red clover, and that was shoots as well as the roots, in SRC treatment as compared to the fertilizer treatment and that alone. What's kind of interesting too is we saw quite a bit of nodulation and active fixation going on with the legumes, both of them, which is not shown here, but that has a lot of implications for the nitrogen potential that would translate into the soil once they were implemented in the field, because Spanish River carbonatite is a mineral, has a lot of trace minerals, and not so much with nitrogen. You don't get nitrogen out of that. So we need some other source that can help us with providing nitrogen to our plants. So in the summer of 2017, we brought everything into the field, and what you're looking at here is a plot map of what our plots look like. So 4 meters by 6 meters, spanning over 3 different, where you see the circles, panels of vines. And the green area would be where you would normally see cover crops implemented in a vineyard setting. So we have a buffer zone to account for any kind of overlap. So when the farmer's doing whatever it is he's doing to his outside of the plot field, we're accounting for the fact that there's no carryover into our own plots. And what we were looking at specifically here were quite a few different parameters. So we were testing for soil characteristics such as nutrient load, structure, moisture, as well as plant community, biomass, microorganisms, and invertebrates. In addition to this, we were looking at differences in herbivory between the vines in the different treatments. And by herbivory, I mean how much damage was being felt by the plants, both the vines and the cover crops. And we looked at the end of vine and grape performance, because at the end of the day, this is still a business being operated by a farmer, and they are very interested in whether or not these techniques are going to translate into some sort of profit. So just to give you a very brief idea of what we have found so far in the field, I should say that there is a ton of information and we are still slogging through it all, but just to give you a little picture of what we found in the first season, we did have consistent soil parameters. This is surprising and important because I'm not sure how many of you are known soil ecologists, but soil is often super variable throughout these areas. So to have consistent soil parameters initially in all of our plots was pretty exciting. And we also expect this because it takes some time for these things to, like the amendments, to actually show any kind of response in a system. So here you're looking at the pH levels, and baseline, midpoint, and endpoint are the 3 collection points in the summer season. So we had May, July, and September. And it's all fairly consistent. We did have a dry year. When you're looking at pH, we're also looking at a graph that would be similar for moisture and for all of the other nutrients, nitrogen and whatnot. And it was a very dry year too, so you see the decline in pH was reflected also decline in moisture. So because nitrogen can be highly variable in soil, and it's also not, I think a lot of people would argue, a really great way to test nitrogen availability to plants We looked at vine petioles because the tissue in the plant often shows a more robust and more accurate store of nitrogen. And in that case, we did find that nitrogen percentage was fairly consistent across all plots, all vine petioles. Again, this is a perennial system, so it does take time for anything to show up, if anything is going to show up. But having consistency, I can't stress enough, was important for our first year. So we saw the same thing in community vegetation diversity quantification as well, which it is a little surprising because we're looking at the difference between ryegrass monocultures and what we have done to increase diversity in the intercrops. However, we did have a few issues with management techniques, and the mower was out a couple of times, and we've since mitigated this potential problem going forward, and we're hoping that we will see some differences this season that we're in right now. The other thing is we're grouping everything into functional groups rather than just species. So once we do get to that point, we do expect to see differences, obviously, because I can tell you right now, just by being out there and seeing, there are a lot of differences in what is there. So there may be the same diversity in numbers, but what is there is going to make a huge difference in the implications down the road. So what was different and very cool, our partners at Laurier took a lot of these soil samples and looked at colony-forming units of heterotrophic bacteria in the soil. And what you're looking at here are two different groups. The first three boxes are from early summer extraction, and the last three boxes are from later in the summer. So the purple represents the plots that, represents the plots that had no amendment put in them. The blue is the pre-fertilizer versus fertilizer, which I guess it's not very blue there, but the second purple is fertilizer. You can see there was quite a drop in microbial activity in the second box in the later summer. But if you look at the SRC, the green box, you see that there was actually an increase in colony-forming units of microbes, which is kind of cool because microbes will respond quicker than a lot of these other components in the system. So I guess what we're looking at going forward are several more questions. The first of which is, will changes in soil microbial community lead to changes in plant invertebrate communities in season 2? And will plots containing intercrop cover crops and SRC rather than monoculture cover crops and chemical inputs support more robust agroecosystems? So that's it for me for today. I just want to thank you for listening to our very preliminary project. And if there's any questions, if there's time for questions.